Uh, Good morning, everyone. I am Rahul Nikam. I am a pediatric neuroradiologist at AI DuPont Hospital for Children. And today I'm going to talk about our project MR Elastography in characterization of pediatric gliomas. This project is done in collaboration with Dr. Johnson's lab at University of Delaware. So looking at the demographics, brain and CNS tumors are the most common type of cancers in children and adolescents with an average incidence rate of 5.57 per 100,000. Embryonal tumors and pilocytic astrocytomas are the most common before 9 years, and gliomas grade 2 to 3 until up to 19 years of age. And imaging plays a very valuable role in not only diagnosis, but treatment as well as monitoring of patients with pediatric brain tumors. One of the major drawbacks of imaging is it has low specificity, as well as there is no good histological correlation. Additionally, the advent of newer targeted chemotherapies and radiation therapies have resulted in very complicated imaging findings, such as pseudo-response or pseudo-progression, which are not adequately assessed with conventional morphologic imaging techniques such as MRI. And this creates that void, or this creates that need for newer sensitive imaging technologies. So if we look at the approach to treatment for gliomas, which was before a few years and what is it currently, so now, according to newer WHO classification, the gliomas have been further subcategorized based on their molecular or genetic abnormalities, and the treatment varies depending on what the underlying genetic abnormality is. So recent advances in genomic analysis have led to many breakthroughs in understanding of this biological features and further targeting various molecular pathways as far as treatment of this tumors are concerned. So I'm just going to introduce few of the favorite terms of all imagers in the world currently. The first one is radiogenomics, which essentially means finding out relationship between imaging phenotypes and expression of gene mutations. A basic example of radiogenomics will be a medulloblastoma. So we can actually ascertain the underlying molecular subtype of medulloblastoma depending on where the location is. For example, if the location of the tumor is in lateral cerebellar hemisphere, it is suggestive of sonic hedgehog subgroup of medulloblastoma. Radionomics takes this whole idea one step further, and it aids in uncovering hidden quantitative data within a pixel or voxel and further correlating this data with other patient-specific biomarkers. Mechanogenomics is a recent term where we can use biomechanical information, for example, data acquired from MR elastography, which we are going to talk in next couple of slides, and correlating this information with the underlying genetic abnormalities. What all these newer approaches does it holds a great promise to an inexpensive, non-invasive phenotyping of tumors, which can be used in individualized patient therapies or treatment strategies. So as we know, according to the recent WHO classification, now gliomas have been further subcategorized based on their underlying genetic abnormalities. For example, in low-grade glioma, BRAF mutation is one of the commonest mutation in pediatric low-grade gliomas. Similarly, high-grade gliomas have been further subcategorized based on histone mutation, IDH mutation, or histone or IDH wild type. Ependymomas can further be subcategorized based on imaging findings and genetic abnormalities. For example, a posterior fossa A subependymoma will arise from the lateral recess as compared to posterior fossa B subtype of ependymoma, which will arise from the midline obex. And one of the very classical example of this is molecular subgroups of medulloblastoma. So the three main imaging phenotypes that can help predict the molecular subtype of medulloblastoma is their location, enhancement pattern, and presence and absence of metastasis. And we can, based on these three imaging phenotypes, we can fairly specifically characterize medulloblastomas in one of the four molecular subgroups. So moving on to MR elastography with this background. So imaging technologies which are capable of offering virtual histology, they have the best prospects of non-invasively detecting and invasing neuropathology. And MR elastography is one such novel technique which helps in evaluating the viscoelastic properties of the tissue. And this viscoelastic properties, they heighten the sensitivity to microstructural tissue integrity. Viscoelastic properties measured with MRE reflect critical information about distribution and organization of neurons and axons, glial cells, and extracellular matrix, giving us insights into the histopathology of neoplasm or any disease of the brain. Before we move on to our data, I just wanted to discuss this paper, which was published in 2018 from the Mayo Clinic group. And in this paper, they tried to determine the relationship between glioma stiffness and IDH1 mutation status. Now, isocytide dehydrogenase gene mutations are recognized as a key genetic prognostic marker for diffuse gliomas. And tumors with IDH wild type tend to behave far more aggressively than tumors which demonstrates IDH1 mutation. So on MR elastography, what they found was the gliomas were softer than the normal brain tissue, and glioma stiffness decreases with increasing tumor grade. And when they compare between the cohorts of wild type versus IDH1 mutants, they found that wild type IDH1 tumors were significantly softer than mutants. And their conclusions were glioma stiffness may be a biomarker of IDH1 mutation status, underlying genetic abnormality, with softer tumor being indicative of wild type IDH1. What is the most important and very interesting fact about this paper was they found that the grade 3 gliomas with wild type IDH1, tumors which are going to do far more worse, were comparable in stiffness to grade 4 tumors than the comparable grade 3 tumors which were IDH mutated. So which means that biomechanical properties of tumors were more correlated with the underlying genetic abnormality than the histopathological grade of the tumor, which I thought was very interesting. So there are no studies evaluating MR elastography in pediatric brain tumors, and our aims for this study was to compare the stiffness of glioma with stiffness of contralateral uninvolved white matter, and to determine the relationship of tumor stiffness with tumor grade, proliferation index, and genetic mutations, and to determine the relationship between tumor stiffness, diffusion-weighted imaging data such as ADC values, and tumor grade. So looking at the MR elastography technique, so it essentially has 3 steps. First step will be introduction of shear waves by applying external vibration. Second step will be the measurement of resultant displacement using phase contrast pulse sequence. And final step will be estimation of the actual stiffness of the tissues using inversion algorithm. So the shear wave generation is done using a pneumatic system, which is essentially a soft pillow-like driver through which external vibration at 50 Hertz is delivered. For data acquisition, a single-shot echoplanar imaging is used. These are the typical imaging parameters. Our sequence lasts for around 3 minutes and 15 seconds, and we usually acquire our MRE data in conjunction with the clinical scans which the patient is going to have. It is just an add-on sequence which we perform at the end of the study. The important concept over here is a longer wavelength reflects faster wave propagation in the media, which indicates higher elasticity of the tissue. The MRE data acquired are input into the nonlinear inversion algorithm, and it returns a viscoelastic complex shear modulus from which stiffness can be calculated. Now, this wave information is used to process 2D color-coded elastograms, and quantitative grayscale maps from which ROS can be drawn and stiffness can be calculated. So this is one of the examples. This is a 16-year-old girl had new onset seizures. We found a tumor in the anterior and medial temporal lobe. It's very hyper intense on T2 weighted imaging. These are just the raw data which is sent onto the PACS curl wave images. But essentially, importantly from this data, we calculate this elastograms. These images are flipped right to left. So this blue blue area which we are seeing in the anterior and medial temporal lobe represents this tumor. And as you can see, this tumor has decreased stiffness as compared to the surrounding white matter. So we'll just look at a few cases quickly. So this was another. This is one of our first patients present with new onset seizure. We found a mass in the trigonal region, which showed enhancement. There was clearly no restricted diffusion. And on stiffness maps, what you can see that the tumor is very heterogeneous, has areas of decreased stiffness. Second patient. We just saw this image before. So tumor in the anterior and medial temporal lobe doesn't enhance, however, shows reduced stiffness on elastograms. And this is a third patient presented with headache, had a large mass in the suprasellar region invading into the cerebral peduncle. The tumor is very heterogeneously enhancing, doesn't show any restricted diffusion. And again, on stiffness maps, there is this heterogeneity within the tumor, and there is decreased elasticity as compared to the uninvolved cerebral hemispheres. So looking at the numbers, the stiffness of the tumor tissue was less than the area which was immediately surrounding the tumor or uninvolved area. And this was true for all the three tumors which we imaged. And when we try to compare this tumor stiffness, now if we look at few of the papers which have been published in adults, the tumor stiffness of something around 2.3 and above will correspond to a low-grade glioma, and all these tumors are around that same range. So this was 2.55. It turned out to be a pilocytic astrocytoma grade 1. This was 1.99 or almost 2. It turned around to be low-grade glioneuronal tumor, again grade 1, with a low Ki67 index. And the third patient who had a tumor stiffness of 2.48 turned out to be a ganglioglioma, WHO grade 1 with a low proliferation index. So essentially, three tumors, three different locations, different imaging subtypes. Somewhere one of them showed no enhancement, other showed enhancement. But essentially on stiffness maps, or the values which we found were consistent with a grade 1 tumor. So our conclusions for this project were, it's too early. We don't have any conclusions. There are a lot of work needs to be done, but we will have our results next time if we are given an opportunity to present. I would like to acknowledge the Delaware and Bree leadership, Dr. Akins, Dawn, and Erin for being a phenomenal help. And their mentorship. I would like to acknowledge my colleagues and my mentors, Dr. Johnson, Dr. Chaudhary, Grace, for doing a phenomenal job processing all the data, and rest of my colleagues. We can be reached at this email addresses. Thank you. Any questions?